Okay, the presentation that I am using is the one right off of eCampus to go over the anatomy of the microscope, and then we'll talk about the different types of microscopes. So the microscope that you're going to use in here is a compound light microscope. A compound light microscope is the one that we are using in here. These microscopes require some type of electric source, so that means you just plug it in. Okay? So once you have it, you have your compound light microscope. It is considered a light microscope because it uses a light for its source, compound because of the number of lenses that it uses in order to focus on your specimen. When you go through, let me preface this. I'm going to go over the anatomy, and then your quiz that you have, your demonstration. You'll have one quiz where you're labeling it and answering questions. The second that you will have will be a demonstration of how to use the microscope. Those of you who stayed, I think it is incredibly intelligent that you did, because even if people say, oh, I really know how to use this, a lot of times there's a struggle there. You have 3 minutes to take your specimen and focus it correctly, and you get scored on that. So once I go through this today and we kind of become familiar with it, hopefully my goal is that you become more comfortable. You guys also have new microscopes, so there is a harness here that you take this off. This is just a spacer to keep this separate. So if yours is engaged, will you please remove it, because it won't allow your stage to move like yours. Yeah. And then whenever you go to put it away, please make sure you put it back. That you put it back. Okay? So before we go any further. All right. When you come up here, a lot of students are like, oh my gosh, the microscope isn't working, it won't turn on. You've plugged it in. The next thing that you should have to do is turn it on. So there's an on-off switch at the back of that microscope. Okay, there's an on-off switch at the back, and just click that, and it should turn your microscope on. The next complaint I hear is, I've plugged it in, and I've turned it on, and the light doesn't work. This is broken. It is not broken. On the right side of your microscope, at the bottom, on the right side, at the bottom, there is a structure called a rheostat, R-H-E-O, rheostat. And it controls the amount of light that is used with your microscope. If you are someone who is sensitive to light and it triggers headaches, you can use a soft light. And if you're somebody who wants a really bright light, then use a lot of light. Okay? You manipulate that, and you can change that back and forth so that you can see your specimen. Okay, so you've turned it on, and you've used the rheostat to change the amount of light that gets through. Going over basic anatomy, and I'm just going through the demonstration PowerPoint that was provided to you on eCampus. Okay? You have the base of the microscope, that's this bottom part. The rheostat is here to control the light. This part right here is considered the arm of the microscope. So if you transport your microscope, we ask that you grab it by the arm and then another hand under the base. Okay, so by the arm and base, you have your rheostat. What else is on this one? Oh, here at the bottom, where the light comes through, it's called the illuminator. Why do we call it the illuminator? Because it illuminates. It's light. You could also call it the light source. I'm not going to get upset either way. They both function for the same thing. Okay? So the illuminator is that source of light. Questions so far? All right? Now, on the left side of your microscope, left side, there is a knob beneath the stage, not the focus, but there is a knob, a little black knob, and when you turn that knob, the structure beneath the stage will move up and down. This knob is called the knob for the condenser, or the control for the condenser. And the structure that is moving up and down is called the condenser. Okay? So you have the condenser knob or condenser control knob, and you have the condenser moving up and down. During a normal setting, you're really not going to mess with this. But I want you to know what it is. Does anybody know what the word condense means? Like you bring it all together, right? So the light is scattered, and when it hits the condenser, what does the condenser do to it? Brings it together to shoot it through your specimen. Okay, so that's what the condenser does. And in normal setting, like I said, you're probably not going to have to manipulate that at all. That knob is on the left side, and it moves that condenser up and down. Okay? On the right side, there are two more knobs. Under your stage, not your adjustment knobs. Not your adjustment knobs, but these two knobs here. The top one, when you roll it, it moves your stage forward and backwards. The bottom one, when you roll it, it moves the specimen right to left. It doesn't move the stage, it moves your specimen right to left. These are the stage controls. So you have the controls for the stage on the right side, and you have the condenser control on the left side. Everyone okay so far? Touching it, putting your hands on it? You're good. Okay? Now, using your condenser control knob, may you drop your condenser all the way down. Using your condenser control knob, may you drop your condenser all the way down. Okay, so turn it all the way down so that you can see it clearly here. When you're looking at your condenser, at the very bottom of it, there is a set of numbers that say max and min, or it may not be numbers, it may just be lines, markings. And then there's a lever that moves back and forth. It's at the very bottom of that condenser. The condenser is actually the part on top. The part you're touching right now with the lever is called the iris diaphragm. The iris diaphragm controls how much of the light actually makes it through the condenser. Does anyone know what the iris is in your body? Yeah, it's the colored portion of your eyes. And when there's a whole lot of light, the iris gets small because the pupil, when there's a whole lot of light, the iris gets big because the pupil dilates, because your eyes don't want a whole lot of light. And when there's not a lot of light, your iris gets small. I said dilate, it means constrict. I'm listening to myself talk, I apologize. When the iris gets thicker, it constricts, going to a pinpoint, meaning we don't want a lot of light there. Your eyes do not want a lot of light. It's an assault to your eyes. When it is a dim setting, our iris will relax and get smaller, and our pupil gets bigger, allowing for us to take in more light or collect more light from that source. This iris diaphragm, the lever for it, controls how much light will get through. So if you bring your condenser back up, leave your light on at a brightness that is comfortable for you, and you look through your oculars, you can move that iris diaphragm back and forth, and you can kind of see how it's changing the amount of light that gets through. Truly not doing it, it doesn't help you to figure it out. So just try moving that iris diaphragm back and forth so you can see how it's changing the light as it goes through. The amount of light, not the light. The amount. Okay? Questions so far? Fantastic. Okay. Now our stage. So this black large portion is the stage. If you're my age, around my age, or you had a high school that didn't have a whole bunch of funds, you did not have mechanical stages. You had two stage clips, and you stuck your microscope slide underneath it, and if you wanted to move your microscope slide, you put your fingers on it and you just moved it. And if you moved it too much, you messed it up. But you guys have these super nice binocular compound light microscopes that have a mechanical stage. I call it the claw. Okay? You can call it whatever. But on your stage, which is the black part, there should be a silver setup. And some of you have it's like a brushed flat black on some of the newer microscopes. But you have this silver setup, and that's our mechanical stage, the claw. How it works, for those of you who are older, you don't force your slide underneath it. This mechanical stage was designed to hug your slide. Hug it. So you open your stage, your mechanical stage, you place your slide in there, in that top right corner, and when you release it, it holds your specimen snugly. Okay? When you go to practice, we have slides already over here that you are welcome to use. Some of you may have brought your blank microscope slides, and you can use those just to practice using this microscope. But you open up that claw, you place the slide in that top right corner, you release it, and it gets that slide in a snug hold. That now, when you use these stage adjustment knobs on the right, you actually move the slide. So if you begin to use your adjustment knobs, you will see that. Okay, so that's your mechanical stage. Then you have these large knobs, and these are the knobs that everybody always wants to touch first. Okay, because I guess they just have that appeal to them. They just say, grab me. These knobs are responsible, the ones that are more medial, the large ones, we call them the coarse adjustment. Before you start playing with it, will you please take your nose piece and put it on the smallest objective, so engage the red one. That way, while you're playing with it, you don't break anything. Okay? So we'll get to where we change them, but right now, if you'll just put your smallest objective, the red one. Yes, thank you. So the red one should be the one that's engaged. When you use the coarse adjustment knob, the one that is medial, the larger one, it moves that stage up and down. Physically moves it. When you stop using the coarse and you use the knobs that are on the outside or more lateral, those are called the fine adjustment knobs, the stage doesn't move. Because these fine adjustment knobs are responsible for fine focusing, kind of like a camera would do, just to kind of get the details of it. So it's not going to physically move the stage, okay? Not going to physically move the stage. So you have your stage, your mechanical stage, coarse adjustment that moves the stage up and down, and fine adjustment that fine focuses it like a camera. All right? Questions? So you have two quizzes. That's a great question. You have two quizzes. One quiz is going to be about the anatomy. One quiz is can you use it? So yes, you will have to know the names. This PowerPoint is accessible to you in your course shell. And so you can go over it. And then, of course, I'll post this video to going over it again. And I 100% believe that you are doing yourself a huge favor by actually touching it and manipulating it. It will make it stay inside of your brain. And then when you take your quiz, I will allow you to have your microscopes out. So those of you who do, you're a tactile learner, you can and be like, okay, that is this. Sometimes it just helps. But you will have a quiz over both. Now, when you're looking at your microscope, you have these two structures here, which are called your oculars. You guys have fancy microscopes, so we have binocular oculars. And they have addressed the fact that some of you have eyes that are very close together. Some of you have eyes that are very far apart. So you have the ability to stretch these out and increase or decrease the interpupillary distance. What? Okay, so you just push them in or out to move that. You don't have to unscrew it, you just push them in and out. Okay? These magnify on their own 10 times. This structure right below will turn. And we call it a revolving nose piece because it revolves. All right? And attached to that revolving nose piece are your objectives. We're going to go over them from smallest to largest. The smallest objective has a red band around it. Smallest objective has a red band around it. And it has the number 4. Depending on where you are studying, we either call the smallest objective the scanning power, or we call it the low power. I am not going to force either one on you, but you need to know both. Because if you go, when I was at A&M, it was 1, we said scanning, and at UT, it was low power. It just depends on who you're with, and you need to be educated enough to know that if they're saying scanning, they're not talking about the low power one. Okay? This magnifies, it tells you 4 times. So using this objective, it's magnified 4 times. Along with what the ocular does, which is 10 times. So the total magnification would be what? 40. You do the 10 times the 4. Okay? That is on scanning or low power. If you roll your nose piece over to the yellow one, with the yellow band, it has the number 10. This is what we normally call our medium power objective. Okay? It magnifies 10 times. My oculars also magnify 10 times. So any specimen viewed on the medium power objective is magnified 100 times. Fantastic. Okay? From here, we go to high power. The high power should have the blue band. The blue band is accompanied by the number 40. 40 times it magnifies on its own, plus times 10 times what the oculars do. So it's a total magnification of 400. The last objective is white. And it says 100. This is called the oil immersion objective. When it is engaged, it magnifies 100 times. Then the oculars also magnify 10 times, so it's a total magnification of 1,000. You will not use this objective in this course. Okay? So when I ask you to get it on high power, I just want it on blue. All right? Now, I want you to please put your low power objective in completely engaged. Completely engage your low power objective. So the red one is engaged. Once you do that, may you raise your stage all the way up using the coarse adjustment knob. Raise your stage all the way up. This part is not in the notes, but I want you to see what happens because it's on both your lab and lecture exams. And this is just an extra bit of information that will benefit you. So, do you see the distance between where the low power objective is and your stage? So about how much room is that? Like, show me with your fingers. So it's about like this much, right? We call that the working distance. Whenever there's that much room, that means within that gap, I can see this, like this much. Okay, so it's kind of shooting down. My working distance is small, so I can see this small field. We call it a field of view. Go from low, please, and thank you, without moving your stage, Just revolve your nose piece to medium, to 10. Now look at your working distance. Show me on your fingers. Did it get bigger or smaller? Because smaller. I got closer to my specimen, so my field of view got smaller, and I'm going to get more magnification. So I got closer, my field of view got smaller, I will get it better magnified. Go from medium, please and thank you, to high power, which is the 40, the blue objective. What does your working distance look like? It's very, and if your microscope slide is there, no exaggeration, it's usually right on top of it. Okay? What happened to what I can see? Did it get bigger or smaller? It magnified it larger, but the field of view got smaller. So my working distance decreased. It has you do this in the lab, but I wanted you to see it because it's something that people struggle with. Okay? So I wanted you to see that working distance. You know the magnification of each of the objectives because it's printed on there. So when you're taking your exam, your quiz, you can reference this. Like, it's not a trick. Like, if you know what you're looking for, you'll be just fine. Okay? Questions right now? All right? So this goes through the powers. So I've already kind of gone through that. And the oil immersion is one that you will not use. You will not engage the white objective. Okay? It's showing you that it's pointing towards the magnification of those objectives, which you and I discussed. All right? Now, over here is our dissecting microscope. This dissecting microscope is a microscope that you are going to have to be familiar with. You will use it in the lab, but this is not a microscope that uses microscope slides. This is a microscope you use, and it's set up differently, okay, when you're looking at something that is not microscopic. So why is it called a dissecting microscope? Normally, we use this when we're dissecting specimens. Like, for example, if you're a medical examiner and you pull out some intestine and you notice some irregularities, in order to use one of these microscopes, I have to take that tissue and make it into a microscope slide. That takes time. But if I have a dissecting microscope, I can come over here, take that whole section of intestine that I wanted to look at, and I could put the whole thing underneath it and look through it, and it will give a closer look. This is not intestine here. Right here, this is a mouse skull, and you'll have it for your lab to look at. And when you look at it underneath there, it magnifies it. You can see stuff on it that you wouldn't see with your naked eye. So these still magnify 10 times, the oculars do, but you can see here that it's magnified 4 times when it's on that setting, and when you roll it over here, it's magnified 2 times. So 2 times 10 would be 20, 4 times 10 would be 40. It only has the coarse adjustment to move it closer to your specimen. So this is a dissecting microscope. Please don't come over here with your microscope slides. Okay, I see a lot of students do that, especially during the lab. They bring their microscope slides in, and they're like, I can't see anything. Yeah, because you can't. This is used for stuff that's macroscopic. All right? The other microscopes that you need to be familiar with are the transmission electron microscope. The transmission electron microscope is one that we don't have on this campus. They usually run around $250,000. What they do is you take your specimen and you coat it with metal, okay, and then you shoot electron rays through it. So transmit means to like go through. So we're transmitting electrons through our specimen. This allows us to see things smaller than what we can see with this microscope. So like in this microscope, you will see the nuclei to a cell. Like when you take out your cells in your mouth, you'll see the nuclei. But you won't be able to see the endoplasmic reticulum or the mitochondria or anything like that. When you have this microscope, you can see that. It's just more powerful. Okay, so transmission, it shoots electrons through it, and you still see it in 2 dimension like you see it on your compound light microscope. The final type of microscope you need to be familiar with is a scanning electron microscope. Again, we don't have one on this campus. These are usually microscopes we find on research campuses. This microscope is usually, the setup is about the size of this table. Usually you have to write a grant for them because they're close to $1 million. But what they can do is incredible. They can take things, and like the one that I saw demoed, they took a human egg, a female egg, and a sperm from an in vitro clinic, and they were really close to one another. They were able to stop them, coat it with metal, and you could see it in 3 dimensions. It was incredible, like to see the surface of a human egg and knowing that it's absolutely super tiny. This microscope pulled it up and made it look just super clear and big. It's crazy powerful. It also coats in metal, but you can see in 3 dimension here versus all the other ones that are in 2D. Okay? Very, very interesting microscope, especially for those people going further into research. Okay? You do need to be familiar with the 4 types of microscopes: compound light, dissecting, transmission, and scanning electron microscopes. All right? Now, that's all in the anatomy portion. You need to be able to demo how to use a microscope. So I am going to show you and walk you through how to use the microscope. And I'm going to do it one time really slow, and then I'm going to do it one time fast, and then you'll be on your own on what you want to do. Okay? In eCampus, there are a list of instructions, proper use for a microscope. What I'm about to do is explained word for word right here. I'm just demonstrating it, so you can see it here in writing. When you walk up to me, we'll do volunteers first, but then you might just be selected if nobody volunteers. You walk up to me, your microscope is going to be in disarray. I am supposed to mess it up so that you're forced to follow the steps. You have 3 minutes. It's plenty of time if you practice a few times. It's plenty of time, I promise. It's messed up right now. In order to start off correctly, you need to lower your stage all the way down and engage your low power objective. This is your start point. Okay? So I haven't even looked through my oculars yet. I'm just getting it set up. So my stage is all the way down, and my objective is low power engaged or scanning. I'm going to turn it on. And then I'm going to adjust the amount of light that goes through. What's comfortable for you? Okay, some of you like bright. You don't need to change it when I check it. I'll be fine. I already come to these classes knowing these days I'm going to leave with a headache because some of you really like bright stuff, which is fine. Okay? At this point, you will bring up your slide. I have this slide up here, and I will have a picture that I will show you before we start doing this quiz so that you know what you're looking for. So you're not just looking and find something black and you're like, that's it. No, it's not. Okay? A lot of people find the edge of the glass, and they're like, oh, there it is. It's not that. You will take your slide, and you will open your mechanical stage. You will place your slide in that top right corner. Just set it there, release the claw, and it will hug your slide. I still have not looked through my oculars yet. I'm going to use these stage adjustment knobs on the right side to move my specimen over my condenser. Okay? Now that I feel like it's in a good location, a good position, I am going to begin trying to focus. Some of you like to use both eyes. I do not. I like just using one eye. You do what's comfortable for you, okay? Because I can make it work either way. So I am looking through my ocular, and I'm going to use my coarse adjustment knob to move my stage up until I can see, and I have it right there. I can see it. I am now going to use my fine adjustment just to make it a little bit clearer and then I'm done. If you did the prep work ahead of time like I did, I turned it on, I put it on in front of that condenser, I adjusted the light, this should just take you, once you practice it, it's one of those things where you're like, oh, and it's done. I know that some of you it's going to take a minute, but doing the prep work ahead of time gets you pretty far, okay? So now I have it on low power, or my scanning power, and I can see it clearly. I have to take it to medium power. I do not suggest you stand back like this and switch it. Because what people say is, oh my gosh, it's gone. Like it moved. No, it didn't move. Okay, but if you watch it, you can keep your eye on it. It can't move. It's not alive. All right? So I look at it. I'm looking at it. I'm going to go ahead and move my specimen towards the middle just a little bit so that I know where to look for it. I'm going from low to medium. It is not clear, but all I need to do is use my fine adjustment knob, a full turn, and I can see it. I did not have to use my course. Okay? I just needed to fine focus it with my camera lenses. From medium power, I'm going to go from here to high power while I'm watching it. It is not focused, but I can tell something's there. I'm going to use my fine focus, and there it is. I personally move it towards the middle, especially if I'm going to be evaluated on it, so that the professor has no option but to look at whatever. It's not like off in the corner. Okay? You have to get it to high power, which is the 40. And then you say, hey, I got it. And I say, okay. Then I come and check it. And I'm like, okay, yeah, you got it. Great job. Now what? Then when you're finished, you have to drop your stage all the way down, go to the low power engaged, microscope slide off, turn off your microscope. All of these steps are outlined here. I'm going to do this a little quicker now. Okay? I walk up. It's my turn. I'm going to focus Volvox, which is algae having babies. You'll be excited. I'm going to turn my microscope slide on. I'm going to take my slide, open my mechanical stage, place it in that top right corner, and release the claw. It has now hugged my slide. I am going to use my stage adjustment knobs on the right side to line my specimen up over my condenser. I am in low power. My stage is all the way down. I have not looked at my specimen yet. Now that I have done all the prep work, I'm going to look at my specimen, or at least look through my oculars, and I'm going to use my coarse adjustment knob to move my stage all the way up or until I can see something, and I can see something right now, and it is very clear. I'm going to use my stage adjustment knobs to move my specimen to the middle, and then I'm done on low power. I need to go all the way to high. So I'm going to go from low, I'm going to watch it, going to medium, it's blurry, I'm going to use fine adjustment, and I'm good. I can see it clearly. Like, I'm not faking. Everything I'm telling you is the truth. Then I look through here from medium, roll it to high. I can see something. It is not clear. Fine focus, and there it is. I'm going to move it towards the center so my professor can't be like, I don't see it. And then I would say, hey, I'm done. Can you check it? And then I'll say, yeah, sure. And I'll look at it, and I'll be like, all right, great job. You should see something that is red, and again, I will show you what you should see before we start. So there's no like, it's not here, it's there. When you're done, I'll say, now what do you do next? You drop your stage all the way down, remove your slide, engage your low power objective, turn it off, mic drop, and you walk away. Okay? Any questions? All right.